So historically, molecular profiling of the tumor is performed using a tissue biopsy. However, taking such a tissue biopsy is an invasive procedure, during which potential complications for the patients may arise. A tissue biopsy may not always be an option due to the location of the tumor or, for example, risk to the patient's health. Over the last decade, however, a liquid biopsy has become available. Using blood, we are able to perform minimally invasive molecular analysis, but also longitudinal analysis of a sample. And in contrast to a tissue biopsy, these samples are easily obtainable and allow for serial sampling. So the cell-free DNA, or cfDNA, is found in the bloodstream. When cells undergo apoptosis or necrosis, they fragment and shed DNA into the blood circulation. When a tumor is present, the fragments of the tumor DNA are shed into the bloodstream as well, and this is what we call circulating tumor DNA, or ctDNA in short. These ctDNA fragments are very small and fragmented, so approximately 170 base pairs in length, and they have a very short half-life. That means that if you take a blood biopsy, you have a real-time view of what is going on within the patient. Most importantly, taking these samples, the ctDNA reflects the biology of the tumor and can therefore be used as a surrogate biomarker as well. To be able to detect these mutations, you need a very sensitive detection method, because these ctDNA may arise in only a very, very small fragment of your sample. There are different approaches that can be used, ranging from targeted detection of a single mutation, targeted detection of multiple mutation, even towards a genome-wide approach. Keeping in mind that if the panel sizes increase, you also will impact your turnaround time and the cost. So these are also types of approaches that are used, ranging from digital to real-time to mass spectrometry PCR, towards targeted and shallow whole genome sequencing. I will focus my talk on targeted, specifically hybrid capture-based sequencing, in which we have developed a custom assay using Twist custom panel. This is the setup of my site. I first go into our in-house panel. Moving on toward the automated and optimized workflow we have implemented, technical validation using reference standards, and our bioinformatic pipeline. So first of all, the in-house panel. The reason we chose such a panel is that it's because it's more flexible. You can insert your own different regions that you are interested in and adapt it over time. In addition, towards a commercial panel, it's even more affordable as well. The panel that we designed is focused on gastrointestinal, bladder, and lung cancer, focusing on all the different regions that we know that are clinically relevant. This ends up with over 1,200 probes, covering 117 kb region. In total, at the moment, we have over 65% on target rate. We have 3 million reads that we'd like to obtain for our sample, because we need to sequence so deep to gain specific sensitivity. And using demultiplexing and making a consensus, we end up with a total depth of at least 5,000 X. For clinical sensitive mutation detection, we developed the assay to get at least a 99% sensitivity for detecting single nucleotide variants and indel mutations of at least 0.5% variant allele frequency. In the table you can see the design of the assay. So there are different genes of interest are selected. For some of them only specific hotspot or exonic regions. We added probe for fusion detection, and we added different regions for copy number variation detection as well. So if you want to library prep your sample, we start with the cell-free DNA, which from itself is already fragmented, so it doesn't need any mechanical or enzymatic shearing. Only partial of your fragments, so aiming for at least 0.5% of your fragments, will contain a specific mutation. in this case the C within them, which you can see here. We will perform end repair, A-tailing, and create an overhang to ligate your adapters. These fragments are then uniquely labeled with molecular barcodes from a patient, but also with a unique molecular identifier, or UMI. And these look something like this. You have your sequencing, so Illumina adapters, your UMI adapters, your region of interest, and then also the same reflected on the other side, so that you can perform a duplex and perform sequencing on a NovaSeq sequencer. The UMIs allow for you to be able to track back which is your unique molecular of interest of your starting molecule. So if you perform PCR, that if in case errors occur, you may be able to trace back which are the true mutations shown in green here, or the amplification errors that occur during PCR in red. In technical terms, if you have only one fragment, we call it a singleton. And if you have multiple fragments, so PCR amplifications, we call this a family. To be able to correct for errors, we make sure that the families consist of at least 2 family members. We deduplicate the regions, perform error correction and singleton removal, and we end up with only a fraction of the reads that we started with. But here you can clearly see the specific mutation of interest shown without any artifacts. Moving on to automated and optimized workflow. We fully automate this on a robot, so library preparation and enrichment is done by a robot and not manually. We've added a couple of steps in here that we performed, including adapting the PCR cycles that are used in the pre-PCR, samples, the number of samples that we use per reaction of probes, if we could optimize our UMI adapters, DNA input we use, removing, for example, probes that are efficiently performing, or checking if a manual versus an automated approach makes a difference. I'll go into detail in the first 4 of these things that we did, to give an overview of how you can optimize your own workflow as well. First of all, the number of pre-PCR cycles and the DNA input per reaction of probes. So we started out with checking if we could use either 8 or 10 pre-PCR cycles, or if the total concentration of DNA used in your capture makes a difference. So Twist advises to use 8 samples per reaction of probes, and a total of 1500 nanograms, ending up with 187 nanograms per sample. But we wanted to see if we would add more nanograms, if this would make a difference in the number of products that are amplified. In the graph you can see all these different conditions and the measurements of the family size, singletons and coverage. So going into detail, you can see that the family size reduces when you increase either the number of cycles or the total concentration of DNA. Since we want at least 2 family members, in our case the input concentration of 187 nanograms works best. Looking towards coverage and on-target rate, you here can clearly see that 10 pre-PCR cycles work the best, as they have the highest coverage and the highest on-target rate. Seeing this back in a figure as well might help to understand these data. So on the y-axis we see the read depth, and on the x-axis a specific region. In this case it's sorted from highest to lowest covered region for each of the different conditions that we tested, so either 8 or 10 cycles versus the input DNA. And here you can already see that the 8 cycles have a lower read depth compared to the 10 cycles. Moving on towards family size, with on the y-axis the number of reads that we obtained, you can see that the 500 nanogram samples have a higher number of singletons, or 1 in a family size, compared to the other condition. So in This means we ended up with 10 cycles and 187 nanograms per sample in the hybridization. We also looked into the quality of the UMI adapters. So on the y-axis you see the FASTQ base quality with the number of cycles that we have mapped. And you can see for the R1 and R4 at the arrows where the UMI starts is that we have a drop in quality. This has an influence on the quality score over our bases in the downstream analysis and may result in these regions being dropped out. So we're still looking into this, and meaning that it has an influence on our duplex analysis. So far we can, with high confidence, analyze single UMI analysis, but the duplex still is giving some errors and will need some optimization in the future. And then the last point for the optimization was the DNA input. So how much DNA would you need to perform your analysis, and taking also into account the fraction of your DNA sample that contains tumor. So on the y-axis are the number of mutant reads. If you have more mutant reads, it will naturally be more easy for the annotation software to annotate the specific variant. On the x-axis, you see the tumor fraction from 0.25 to 1%, taking into account the number of DNA, the amount of DNA that is used within your library prep. Naturally, the more DNA, or the higher the frequency of your variant, the more easy it is to detect your mutations. You can see the median number of mutant reads that is picked up within each of these samples, making it of course a bit harder for the annotation software to detect the lower frequencies. But as I said, we wanted to detect at least 0.5% frequency, and we are able to do this with at least 20 to 30 nanogram per sample. Moving on towards the technical validation using a reference standard. Using reference standards they are different availabilities on the market. So one of them is a Circulating sample that I show you here. These are synthetic samples in which different mutations are spiked in different frequencies. So if you sequence them, you know that you need to pick these up to validate your sample. And here are the mutations that should be in the sample for, in our case, lung cancer. A couple of SNVs, Indels, and fusions in different frequencies. So we looked if we can ultimately detect them. We made a difference between single nucleotide variants and indel mutations because we know that the annotation software may have some difficulties with detecting indel mutations compared to the SNVs. On the left, the 1% and 50 nanograms. Towards the right, the lower variant allele frequency of 0.25 and a lower DNA concentration. The annotation has no difficulties with detecting SNVs when there is 50 nanograms of DNA available at 1% frequency of tumor frequency. But as expected, indels are posing a bit of a problem if you have either a lower frequency or a lower DNA input. But again, here we used a variety of DNA concentrations and percentages and are reliably able to detect all of the mutations that are clinically relevant up to 0.5% variant allele frequency. And then the last part is the bioinformatic pipeline. We sequence all of our samples using a NovaSeq sequencer. We deduplicated and removed their singletons using FGBio. Variant calling was done using GATK's MUTEC2. And in our case, we used a panel of normals, meaning that we sequenced at least a number of healthy controls with the same panel and the same material type to be able to remove background errors. Fusion detection was done using GeneFuse, and indels were optimize using an in-house developed tool. So Indels, as I already showed you, are a problem for the annotation software in general. So what we did is we developed this Indel Finder tool in-house, and you give it a number of regions that you'd like to make sure that you do not miss those specific regions. For example, in lung cancer, EGFR exon 19 is one of the most clinically relevant regions. So in this case, the software tells us to have another look at that region because there might be a variant that we may have missed in the annotation. To manually check all of them might be a bit hard, but as you can see in the graph in the picture here, we see a BAM file. You see within the region of exon 19 that there are 8 bases deleted at this specific spot. Zooming in on this region, we can clearly see that there is a deletion. Using this tool, we are able to increase our Indels sensitivity up to being able to detect variants up to 0.25% variant allele frequency. Taking together, of course, you need also to perform some quality checks. So we'd like to use our tool and all of the analysis we do in the end for diagnostics. But we have some quality parameters that we implemented. For example, we will always check the gender. So software will annotate the gender and you have to check it with your patient. A couple of quality parameters are always double checked. And most importantly of those are, for example, the on-target rate and the coverage. If one of those fails, it may result in you yourself missing a variant that is clinically relevant for a patient. Focusing on coverage, we also make sure that the clinically critical regions that we know that are actionably target, that we need to know that are 100% need to be sure that we don't miss anything in there. The software will give us a warning if that is the case. And analyzing your data, you need to pay extra attention to this specific region. And lastly, of course, we will check the insertions and deletions within specific regions. So in the figure again, you can see for which we name the clinically critical regions within lung cancer. And you can see, of course, that there's a high variability within the regions, but still overall we gain a very nice and even coverage for all of our regions of interest, being able to accurately annotate and detect our mutations up to 0.5% allele frequency. So with this, I'd like to thank all of my colleagues that worked with me on this project from the Department of Human Genetics and the Laboratory of Tumor Genetics. This research is done within the Radboud University Medical Center, and the research is partly funded by AstraZeneca.